Session. So we just had the P0, now we have the F1. So we have Mathieu Flamand, and he's a graduate student in Thomas's lab. He's going to tell us about, the title of his talk is Toward an Understanding of Cooperative microRNA-mediated Silencing. So take it away.
Thank you very much. So hopefully with all of the previous speakers, I wanted to do such a big intro. So if I can get the pointer. No, okay, yeah, so there it is. All right, so as you've just seen, our lab has been interested into understanding how microRNAs regulate their target, but more specifically, I've been trying to understand how different microRNAs can cooperate together in order to do this silencing. So as Thomas just explained, there is principally two different mechanisms how microRNAs were shown to regulate their targets, the first one being transitional silencing, and the one that I will be showing more today is deadenylation. So deadenylation was shown to work to recruitment of the CCR4 NOT complex. And how do we see it? Is through our cell-free extract, where we can incubate radiolabeled RNA, and on gel we see that the RNA gets shorter. But we've seen this already. So one of the key features that we have seen in our cell-free extract is that there was a requirement for at least 2 microRNA binding sites to have any sort of deadenylation. So if we use artificial reporters containing miR35, and we have done the same thing with reporters for different microRNAs, we only started to see deadenylation when we had at least 2 or 3 sites. Also, we observed the same thing using natural UTRs, where 2 microRNA binding sites were predicted, and if we mutated either of these sites, in the example of the TO1 gene here, where there's a predicted bantam and miR35 site, mutating either of these sites would slow down deadenylation to almost nothing. So this leads us to the question that I was interested in, which is what is the mechanistic basis of this cooperativity? Luckily for us, using the extract, we found a very useful UTR, which is again was predicted to only have 1 microRNA binding site, but was still deadenylated. And what we found in this UTR is that there was the presence of a non-predicted microRNA binding site or a seedless site, because if we blocked miR35 here, we were able to stop deadenylation, as well as when we used the bantam inhibitor. We could also mutate this site. So basically, we tried to figure out where in this UTR this site was present, and through mutations, we found that the site was present right beside the bantam site, and we could mutate, block deadenylation. We could also restore its seed to enhance the deadenylation, as well as mutate it to a different microRNA, and we still add deadenylation. What was really interesting about this site was its location in the UTR. It was sitting in very close proximity to the miR80 bantam site. So here we can see the seed site with its conserved seed that is binding, and we can also see the miR35 site, which has 2 mutations in its seed sequence. However, in between these 2 sites, there is only 1 nucleotide. And this was surprising because previous CLIP experiment had suggested that there would be a larger footprint of the Argonaute on the mRNA. However, we knew that the seedless site was required for the function for deadenylation, and we decided to use this tool here to look at biochemistry of recruitment of the different Argonautes on the RNA. And for that, we used, again, 2 pyramidal pull-downs, which Thomas just mentioned. So when we do these experiments, what do we observe? If we do a 2 pyramidal pull-down using a miR35 site, we pull down a lot of ALG1, ALG2, because miR35 is the most expressed microRNA in the embryo, at least in early embryos. If we use the sequence of the TO1 gene that has the bantam site, we can pull down a sizable amount of Argonaute. However, the seedless site by itself could not pull down any meaningful amount of RISC, where we can barely see any Argonaute and no AIN protein. And we can compare it to a seed miR35 site here. When we combine the 2 sites together in the same RNA, what we observe is that we had an increase of Argonaute being pulled down, and we can then mutate this seedless site, where we see a reduction. And then if we mutated the bantam site, we lost all pull down of the Argonaute. So this suggests that having a first site present on the RNA can help to recruit a second site, which by itself could not be bound. If we also restore the seed on this sequence, we can pull down even more Argonaute, showing that there's really a cooperative binding of two different RISC on the same RNA. So from this, what we could see was this led us to a first model, which we call transseeding. So basically, we observed that having a first RISC bound on the RNA can help to recruit a second RISC, which has a lower affinity, but it can also enhance the binding of two RISC with high affinity if in the context of the seed restore. However, this model does not explain how the further silencing machinery is being recruited to this RNA. So here we have an enhancement of binding, but not of silencing. But so for this, we did a few more experiments trying to understand how this is done. The first observation came again from the experiment that Thomas just explained, DRIP. So basically here we do the same experiment, but instead of looking at binding on one single RNA, we can compare the binding to different components with target sites that have different number of microRNA binding sites. And what we saw is that the deadenylation machinery, here represented by the NTL1, really is enriched only when there is multiple binding site present. So this suggests that when you have a single miRISC present in the RNA, you don't have recruitment of the deadenylation machinery, and it all only comes when you have a cooperative miRISC present. In order to better understand how this cooperation could occur, we decided to, with our collaborators Hinar Gan and Chris Gonzalez, to model how Argonauts could sit on this specific UTR. So here what we have done is we have modeled the structure of the C. elegans ALG2 based on the structure of the human AGO2, which was solved 2 years ago. And then we were able to dock inside these Argonauts the triplex of the RNA of the TO1 UTR and the miR58 or Bantam microRNA as well as miR35. And then through thermodynamics computations, we could see how these two Argonauts could be placed. So the first thing that we see is that it is possible to have two Argonaute proteins in such in close proximity. They actually seem to sort of spoon together and are in very close proximity. So the second thing that we see from this is that there are potential interaction interfaces here. So two helices at the top as well as loops at the bottom. So we thought that these could be potential interaction interfaces, and we thought that if we could mutate residues at these positions, we could affect microRNA-mediated cooperativity. So for that, we decided to do a screen, and this screen we did it through CRISPR. So the idea was to mutate both in ALG2 and also ALG1 these residues and trying to see if these would have a phenotype. We mainly use ALG1 to start because we have a phenotype on ALG1, and that enables us to do some genetics, which we cannot do easily on ALG2. So here are 4 initial mutants that I will show the results today. So the first thing that we did is we tried to validate these mutants to see are these mutant functional in vivo. What happens? Are they loaded? Are they forming a mature miRISC? So what we can do is we can look if they're expressed. So if we look in L4 animal lysate, we can see that all the proteins are expressed. We can also do immunoprecipitation against ALG1 and look for interaction with the GW182 protein AN1. All except one retain their strong binding to AN1, so one of the mutants did affect this, but at this point we don't know how this interaction is lost, since it is not in one of the GW pocket. And we can also redo 2-parameter pull-downs, and we can see that they are loaded here, these 2 mutants. The next thing that we did is we went on and trying to see if these mutations affect the function of microRNAs in vivo. So we went back to do some genetic assays. The first one being, again, the LAUSI6 assay, which Thomas just talked about, which is based on the biological function of LAUSI6, which is important for the cell fate of one specific neuron, the ASCL neuron. In the LAUSI6 hypomorph, we have a defect in this cell fate of around 20%, depending on which temperature the assay is done. And 3 of these mutants actually showed an increase in the sulfate defect, meaning that these mutants had defects in the microRNA pathway here. Then we can also look at just how these mutants animals are. Interestingly, these same mutants exhibited some vulval bursting. So this phenotype is associated with defects in LET7 through the misregulation of the Lin41 UTR, as was shown by different groups before. And this was also exacerbated when we crossed the mutants in the LG2 null background. In addition, these mutants also have smaller brood size, and here, interestingly, even though 3 mutants had a strong interaction with LASI6, one of them has a really larger effect on vulval bursting. So from these, what we can conclude is that when there is a single miRISC present on the RNA, we do not see any recruitment of the CCR4 node complex. What we think happens when you have first site, you can help to recruit a second site through what we call transseeding. And once you have these 2 Argonautes present on the RNA, we believe that there is interaction between these 2 Argonauts through these 2 interfaces which we have identified. And this would lead to an allosteric regulation of the Argonaute, which would transmit a message through the AIM protein to enable recruitment of the CCR4 node complex. And by disrupting this interface, we think that we can block this in vivo. However, this does not tell us if the recruitment is sufficient for activation. So one other possibility is that there is further regulation and that you could have activation of deadenylation through this allosteric regulation here. So to finish, I'll just like to thank people in the lab who has contributed. So, and as well as collaborators, so Chris Gonzalez and Enar Gan, which has enabled us to have a good grasp of where to do these mutations. And without them, we would not have been able to do that. So thank you.
All right, we've got plenty of time for questions, so fire away, Brent.
Great talk, Mathieu. Does the one mutant that you made that had such a dramatic effect on vulva, does that correlate directly with what you expect to see with deadenylation and so on?
So you mean deadenylation in vitro? Is that what you're saying?
Yeah, exactly. Does the in vivo correlate?
That's the
with
thing,
in
right?
vitro?
So we try to make extract out of these mutants. Deadenylation is slowed down, but it's not blocked completely. So it's either that there are partial mutants, and you would expect maybe if they would like behave as a null otherwise if they would block deadenylation completely.
Interesting that one was not as strong as that one. I forget the number.
So the 2 that have a similar strength are on one side of the Argonaute, and that other one is on the other interface of the Argonaute. So maybe that's the reason. It's at this point, I don't know.
Do you know if having a good seed match can actually help to overcome having a poor seed match? Like in your TOE1 example, if you were to mutate that Bantam site to a miR35 site, which has mispairs in the seed region, would you still get cooperative binding, or does somehow having an Argonaute bound to a good seed match there help to recruit Argonaute microRNA complexes that might have a weaker interaction?
We haven't done any sort of analysis on the Bantam site itself. We have done a lot of mutations on the seedless side, trying to understand how it works. But yeah, we don't know really about position of the seedless site on either side. We haven't done those sort of studies as well. So that's interesting. Could be worthwhile doing.
Okay. If there aren't any other questions, we'll move on to the next speaker. Thanks, Mathieu.